All right, so thinking back to last class period, we finished up talking about the sense of smell, our very first of the 5 special senses. Okay, what we're going to see for the sense of taste is a lot of similarities. Okay, both taste or gustation and smell or olfaction, as it's described, are chemical sensations. That means we're detecting chemicals. In this case, we're tasting chemicals. Okay, and this takes place within structures known as taste buds, most of which are found in little bumps on our tongue called lingual papillae. Now, you can find taste buds in a couple other places, like the roof of your mouth down into your throat, but the majority of our taste buds are right there on the surface of our tongue in these little bumps. Looking at the figure A there on the right, you can kind of see some of those little bumps illustrated. That's a picture of the tongue. Okay, there are 4 types of these little bumps, papillae. The first 2 actually have nothing to do with the sense of taste, and that's because the first filiform have no taste buds. So they're tiny spikes instead, but what they have are tactile receptors. And as we learned the other day, tactile sensations are like touch and pressure. So they are going to allow for you to feel the texture of the food that's on the surface of your tongue. Okay. But since they are tiny spikes, they increase the friction between the tongue and food to help move that food through the oral cavity. So they help with, like chewing and then swallowing. If you've ever felt like the surface of a cat's tongue, like if you've ever been licked by a cat, it feels like what? Scratchy, sandpapery. That's because they have a lot of these filiform bumps. Okay. Foliate also not very important for the sense of taste because they, most of which anyway, degenerate in early childhood, which leaves us with fungiform and valate. Fungiform located at the tips and the sides of our tongue, only contain about 5 taste buds each. And that leaves us with the final type, valate, which basically are these right here. They form like an inverted V at the back of the tongue or near the rear of the tongue. They contain up to half of the taste buds over the entire tongue. So a very small space compared to the rest of the tongue, but very, very important for the sense of taste. Each of them contains 100 or 300 of these taste buds. And here's what a taste bud looks like. It's an oval shaped structure made up of 3 types of cells. Okay, and it's pretty much the same situation as we saw the other day in our nose. You have a receptor cell that is actually doing the tasting. Then you got basal or stem cell, and then you got supporting cells. Okay, let's talk about the taste cells, or as they're more fancily known as gustatory receptor cells. They have a banana shape to them with little microvilli on the end that faces into the crevice called a taste pore. These little microvilli also go by the term taste hairs, and they are the receptor sites for food molecules. So this is where food molecules bind and activates these cells. Now, these guys are unlike the olfactory receptor cells that we talked about last time, because olfactory receptor cells are actually neurons. These guys are examples of what we call, if I go all the way back towards the beginning of the lecture, they're an example of separate cells, specialized cells that actually synapse with the first order neurons. And we saw this illustration the other day. That was actually one of these taste cells or gustatory receptor cells. They become activated, releasing neurotransmitters as a result that then activates and creates actual potentials or nerve impulses in the actual first order neurons. So if we look at our illustration, oops, went too far. Okay. You can see here at the bottom are the actual first order taste neurons that would be synapsing with these gustatory receptor cells. Okay. Basal cells, these kind of purple shaded ones, are stem cells found at the periphery of the taste bud, and they develop and produce what we call our supporting cells. Supporting cells, actually, these kind of reddish shaded ones, will develop into microvilli, or excuse me, not to microvilli, but they contain microvilli, and then they develop into receptor cells. So the basal cells develop into the supporting cells then develop into receptor cells. And that's because those receptor cells only last for a little over a week, about 10 days before they need replaced. Okay. Now, when it comes to taste, there are 5 primary taste sensations. Let you guys write these out. Like most senses, taste requires the detection of molecules. Chemicals that we taste are called tastants. Okay, and a very important component of taste is our saliva. Okay, and you might not think that, but your saliva, your spit, is very important to help dissolve those tastants, and that then allows them to make contact with and stimulate those taste hairs. Okay, a cool example of a way to demonstrate how important taste, or how important saliva is for taste. If we had a lab where we could do this all together, we would do it. But you can just like dry the surface of your tongue kind of one of two ways. You can just like hold your mouth open and just let your breath just kind of dry your tongue as you breathe in and out. Or you just like take a paper towel and dot your tongue until it dries off. Then with your tongue sticking out of your mouth, you can sprinkle a little sugar on top. And what you'll experience is that sugar just kind of sits there. And for a good little while, you won't actually taste that sugar. Whereas if your tongue was nice and wet, you'd taste that sugar almost instantly. Now, speaking of sugar, sugar we all know is sweet. That's one of the 5 primary taste sensations. Sweet's produced by many organic compounds, particularly sugars. Salty, the first one there, is produced by metal ions like sodium, potassium, and we have learned that, like for example, sodium chloride, potassium chloride, those are salts, and that's because they have sodium and potassium in them. Sour is a sensation usually associated with acids. Bitter is produced by alkaloids or basic substances designed, typically causing an animal to reject the food, as in like it's spoiled or diseased. Typically, bitter is not a flavor that people, or a taste that people enjoy. And then I believe this is a Japanese word, if I remember correctly. Umami is a meaty or savory taste produced by amino acids. And as you know, amino acids are in proteins, and proteins are the most dominant part of steak and other meats. Now, when you drink something like Dr. Pepper that I got in my cup over here, or you eat a sandwich or something like that, are you tasting something that's strictly sour, strictly sweet? Probably not. What you're going to experience is more than likely it's a combination of those 5, or some of those 5 anyway. Now, just like we talked with smell, let's kind of describe how this taste process really works. Okay, first of all, once again, it's very important for the tastant to dissolve in saliva, enabling it to make contact with the plasma membrane of the gustatory microvilli. Looking down here at the bottom of the page, we've got 2 taste receptor cells, or gustatory receptor cells. Here are the little microvilli. Those are the hairs, or microvilli, as they are also described. Okay, and that's where you find the receptors for the things that we taste. We can see salty, sour, sweet, bitter, and umami. So those are the 5 primary tastes. The saliva helps dissolve those substances so they can come into contact with the plasma membrane. Okay. Now, no matter how we get to this point at the bottom, what results is the release of neurotransmitters that activate those first order neurons. Okay. In the case of things that are salty and those that are sour, for example, sodium chloride, if it is sodium chloride, it's table salt. Okay, with the sodium ions, there are literally sodium channels within those taste receptor cells that allow sodium to come in. In the case of things that are sour, remember we said sour is associated with acids. Hydrogen ions, H+ is always associated with acids. Hydrogen ion channels could allow those substances to come in, creating a depolarizing receptor potential, and that's what triggers the release of those neurotransmitters. In the case of sweet, bitter, and umami, those involve, just like what we learned with smell, a second messenger system where you've got a series of dominoes, each knocking down the next. But like I said, no matter what, if you're looking here or here, the same result occurs with the release of neurotransmitters that then stimulate those first order neurons. Now, looking on the right side of the page, we show that there are actually 3 pairs of cranial nerves. It really should say 3 pairs of cranial nerves, not just 3. Okay. But you have a pair of each of these that are listed there, which are the first order neurons that innervate our taste buds. They include, and the majority of your tongue is innervated by this one, the pair of what we call our facial nerves. Towards the back of the tongue is where we have the glossopharyngeal pair, and then the very back towards what we call the epiglottis is what's called the vagus nerves, which we know definitely about those. But they're going to transmit signals into our brain, okay, to the medulla, and then into the limbic system, the hypothalamus, or the thalamus. Okay, let's talk about the thalamus first. Okay, remember the thalamus is our gateway to the cerebral cortex, and that's what's illustrated here. Signals travel up to the thalamus, and then from there, they travel out to the primary gustatory area, or the primary taste area, as it's labeled here, gustatory cortex, which is in your parietal lobes, giving rise to conscious perception of taste. And so that's what enables you to actually be mentally aware if it's something salty, sweet, bitter, whatever. Okay. Taste signals are also going to project to our limbic system, which we know is our emotional brain. Good. Okay. And thus will provide us with emotional and memory evoked responses to tastes, which is just like we learned the other day when it came to smell. So we talked about that the other day. Taste and smell actually really similar. In fact, those two really affect each other. If you lose your sense of smell, like people that have COVID, it can really impact your ability to taste. Okay, our next structure that I'm going to talk about, I'm going to start by describing the structure instead of the sense, will be the ears. Okay, and we all know that our ears are the organs that allow us to hear. Okay, but our ears are also very, very important because they contain the receptors that respond to our sensation of equilibrium or balance. So your ears are very complicated physiologically. They're also very complicated anatomically. Okay, and this is an anatomy class, but I think it's important to kind of have a general understanding on the structure of the ear, because the structure always affects function. So we'll start by talking about your outer or external ear, which is the part of the ear that you can see, you can feel, you can get pierced. Okay, includes what we would call kind of like our earlobe, but also the part that we can stick a Q-tip into, what's called the external auditory canal. Function of your outer or external ear is just to help funnel sound waves into the middle ear. Okay, the middle ear is where you find your 3 auditory ossicles. These are tiny little bones, what we call the malleus, the incus, and then the stapes. Okay, your middle ear is a very small cavity in the head. Okay, between the outer ear and the middle ear is a structure called the tympanic membrane, and the tympanic membrane typically is what we call our eardrum. Okay, the function of your eardrum is to transmit sound waves that come with the things that we hear. When they strike the tympanic membrane, they transfer those sound waves inward to what's called the cochlea, thanks to the malleus, the incus, and stapes. If you notice, the malleus is attached to the back of the tympanic membrane, then the incus is attached to the malleus, and then attached to the incus is the stapes, which leads and covers up what we call the oval window, which leads into the cochlea. Okay. Now, what's amazing is the surface area of the eardrum is about 18 times that of this little tiny opening, what we call the oval window, and thus it provides a lot of convergence of that energy to a very small area from a much larger area. So it helps to transfer energy to a very small concentrated area. It's basically funneling it in to a small area. Now, to help prevent damage to the inner ear, our ossicles can at times serve to decrease the transfer of this energy. Okay, and this decrease of energy is called the tympanic reflex. Okay, the ossicles, which you can't see here in this picture, have a couple small muscles attached to them. Okay, and that includes a muscle literally called the stapedius muscle and the tensor tympani muscle. But they reduce the mobility of the stapes. So in other words, they reduce the energy that the stapes is carrying. Unfortunately, this reflex has a latency, so it's a delay of about 40 milliseconds, meaning that it's not quick enough to protect the ear from sudden artificial noises like a gunshot. And that's why if a person is doing a lot of shooting with a gun and they're not wearing ear protection, their ears can be very damaged. If you listen to music that's really, really loud, that can be very damaging. It can cause your ears to ring and all sorts of bad things And that's why as we get older, a lot of times our hearing is pretty diminished. Okay. Now the inner ear is where we transduce. Remember that word transduce or transduction means to convert sound waves into actual potentials. That all takes place in this structure called the cochlea. And we're going to talk about that here in the next couple of slides. After we get done talking about here, we're going to talk about the structures over here, where the cells that create actual potentials related to the sense of equilibrium or balance are located. So let's first talk about the cochlea and the receptor cells for hearing. Now, you may have heard of somebody having a cochlear implant put in. That's people that have trouble hearing. They have a cochlear implant. It's a mechanical device that basically helps to allow people that are hard of hearing to hear. We have a cochlea in each of our ears, which contain hair cells. Hair cells are the receptor cells that have the capability to transduce mechanical vibrations into electrical signals, located within a structure known as the organ of Corti or spiral organ. Okay, and that's found within what's called the cochlear duct of your cochlea. Okay, at the tip of each of these hair cells are about 40 to 80 what we call stereocilia. Okay, little finger-like microvilli, little hair-like microvilli, which are the actual receptors for the sense of sound. Okay, if I go ahead a couple slides, here's what those hair cells look like. Okay, and here's those little finger-like or hair-like extensions. Okay, you can see one's labeled here as stereocilium. That's the singular form, and since only one is labeled, that's why it's cilium instead of cilia, which is the plural form. Okay. These hair cells are just like those taste cells that I just talked about, in that they are not neurons, but they do synapse with first order sensory neurons, which form what we call the cochlear branch of our vestibulocochlear cranial nerves. Remember the vestibulocochlear cranial nerves number 8, we learned in our last unit, are the nerves that supply our ears. Okay, the cochlear branch is the branch that's connected to the cochlea. The vestibular branch is associated with the vestibule, hence the names. Okay, the cochlear branch is specific for carrying information or action potentials for hearing, whereas the vestibular branch is associated with balance. Okay, these first order neurons synapse in the medulla with what we call cochlear nuclei. Okay, some of these decussate or cross over, terminating in the midbrain, and we can see that taking place right up in here. Some of them don't, okay, and they terminate in the pons on each side of the brain. Now, regardless, both signals coming from the pons and the midbrain are conveyed up to the thalamus, and then the final signal is then propagated to the primary auditory area of the cortex and your temporal lobes, and we can see those things are labeled there. So how does it all work? Well, first, the sound waves must reach our auricle. Auricle is just a fancy word for your external ear, the actual part of your ear that you can like see, feel, and pierce, what people typically call their ears. Okay, sound waves get transferred inward, striking the tympanic membrane, causing it and the malleus, the incus, and the stapes to vibrate. Okay, so just like a drum, when you tap a drum, it vibrates. Your eardrum or tympanic membrane vibrates, and in doing so, that causes the malleus, the incus, and the stapes to all vibrate because they're all connected. Okay, so it's just like a chain reaction. This causes the membrane of the oval window to vibrate, and what results on the inside of the cochlea is what we call perilymph, which is fluid, and endolymph, another fluid. Those move. Okay, and I always think of this like the scene in Jurassic Park, which let me see if I can find that scene for you. Sometimes students don't know what I'm talking about. So in this iconic scene of Jurassic Park, I don't even need the audio, but I'll turn it on nonetheless. You can see these kids are sitting in the vehicle and the water is vibrating. Okay, what's happening out in the distance is a T-Rex is approaching, and so every time the T-Rex steps on the ground shakes, and therefore the water shakes. Okay, Those are called impact tremors, and I'm going to stop it here because you get the idea. But when the eardrums vibrate, that transfer of energy is sent into the cochlea, causing a ripple effect, just like what you see here with this water. Okay, now it's probably not perfect like this, but hopefully you guys get the analogy. So that's what is taking place inside the cochlea. Okay, and this movement of the endolymph, that fluid, causes the basilar or spiral membrane to vibrate, and that's what stimulates the hair cells in the organa corti to trigger and transmit nerve impulse along the cochlear branch of the vestibulocochlear cranial nerves. Okay, now sound has different pitch. Okay, when you think about people like in a music group or in a chorus, you know, you have like the tenors, what is it, altos, who are the people that sing real low? Bass, I don't know. I'm not a music guy. But the different levels of pitch relate to the sound frequencies. Okay, higher frequency waves are going to stimulate hair cells near what we call the round window, whereas lower frequency, that would be deeper voices, stimulate hair cells near the beginning of the cochlea. Okay, and that's why when you stimulate those, you're able to process that in your brain as, and you can tell, like if I'm speaking with a really high pitch versus a really low pitch. Okay, you're transmitting those signals to different areas of the primary auditory area. Okay, if you guys remember the other day, we talked about like the homunculus, that little doll thing that I showed you, and how like we carry signals from our feet to a specific part of the cerebral cortex. Well, it's kind of the same thing here. High frequency sounds are transmitted to this area of the primary auditory area. Lower frequencies over here in purple, and then medium frequency is here in red. So that's how we're able to determine and decipher different frequencies. Okay, so here's just kind of the pathway. This is kind of showing you the middle ear, this structure over here. It's kind of an illustration of the cochlea. Here's our sound waves hitting the eardrum, the tympanic membrane causing the malleus, the incus, the stapes to all vibrate. And then the ripple effect occurs on the inside of the cochlea, okay, which bends the hair cells. And the bending of those hair cells triggers a receptive potential and thus triggers nerve impulses. Once again, keep in mind these, you know, all these different steps, not going to be something I'm going to ask you on the exam. And I'm just making sure you kind of know the main things, especially you want to make sure you know like the vestibulocochlear cranial nerves are involved with hearing, okay, specifically the cochlear branch. Last slide here, just want to talk about transduction, how we stimulate those hair cells. Okay, they transduce or convert mechanical vibrations into electrical signals, and it involves those little finger-like extensions called stereocilia. Okay, at the tip of those stereocilia, you find the receptors, okay, and connecting the stereocilia to one another are these tip link proteins. Okay, now notice here that, and it's not on accident, that these stereocilia are of unequal size. It goes basically from tall to shorter, from left to right here. Okay, if we look on figure B and figure C, the hair cells can bend their stereocilia towards the bigger side, or they can bend in the other direction toward the smaller size. Okay, well, if they bend in the direction of the taller stereocilia, what happens is those tip link proteins open up the ion channels, allowing things like potassium that you see here to come in. Okay, this causes a depolarizing receptor potential, and that triggers then therefore the release of neurotransmitters to activate those first order neurons. Okay, but if the stereocilia bend in the other direction, those tip link proteins basically close off those ion channels, and therefore no neurotransmitters are going to be released. And so we would say this is a hyperpolarization, and that decreases the frequency of nerve impulses. So this neuron is less likely to fire action potentials to be propagated into the brain. Okay, and once again, this bending of the stereocilia is a direct result of that ripple effect of the endolymph, the fluid, like that water in the glass in the scene that I just showed you. Now, the wonderful thing about equilibrium or balance, a lot of similarities to what we just learned for hearing. Okay. Defining equilibrium, it's the ability to maintain coordination, balance, and orientation in a three-dimensional space. I always tell students to really appreciate your equilibrium, your balance. I would say between the five senses, this one is the one we take for granted the most. Okay. And that's because, you don't think about having balance until you don't have balance. You guys ever heard of vertigo before? We feel dizzy all the time. Okay. I was just talking to one of my fellow instructors, and she said that a few years ago she went on a plane ride, and I don't know if something popped in her ear or what. She might have had an infection or something. But for the next month, she said she felt drunk all the time. And not the good part of being drunk, but the part of being drunk where you're spinning, you're nauseous. Can you imagine what it's like every day for that long time? Think about how terrible life would be if you were experiencing that, not just for a month, but for long periods of time. Okay. Equilibrium is absolutely important to be able to get by and not go insane. Receptors for this equilibrium process are hair cells, just like with the sense of hearing. They're found in these three semicircular ducts and two chambers, what we call the anterior saccule and the posterior utricle. If I go back to our picture of the ear, here are these semicircular canals or ducts, and then the utricle saccule stuff is kind of down in this area. Okay. Now, there's two types of equilibrium, static and dynamic, which we've seen those words before. Static, think stationary. And if your body is stationary, it means you're not doing what? You're not moving, okay? So this is the perception of the orientation of your head when your body is stationary. Accomplishing this is a structure called the macula, okay? The macula sacculi lies vertically on the wall of the saccule, and the macula utriculi lies horizontally on the floor of the utricle. Changing your head position, like when you bend over or when you turn it to the side, changes your pressure, and thus stimulates hair cells, in turn stimulating receptors of the vestibular nerve. And we'll see pictures kind of illustrating this on the next page. Much more complicated is dynamic equilibrium, which is the opposite of static, in that it is equilibrium when you're moving. And then so it's the perception of motion or acceleration, which can be linear, like going straight ahead, or much more complicated, like when you're spinning, like Simone Biles when she's doing her triple flips and spinning, that's angular. Okay. Linear acceleration is perceived by the macula, angular is perceived by the crista. Okay. The transduction of equilibrium is basically just like what we saw with sound transduction. You have hair cells with stereocilia, and they bend in certain directions. Okay. And so it's basically the same thing. So I'm not going to get into great detail on it because we just did it for hearing. But they depolarize, hyperpolarize in a similar way to the hair cells in our cochlea do for sound transduction. These guys synapse with the first order neurons that form the vestibular branch of your vestibulocochlear nerves. Okay. Looking on the bottom of this next page here, we have the utricle saccule stuff that I was just mentioning illustrated. Okay. Looking, if you can see, it's kind of small, but these are the hair cells, and then you can see the little blue lines here. If we zoom in at them, those are the stereocilia. Okay. Now, covering those hair cells and the stereocilia is this structure called an otolithic membrane. Okay. Think of this like a pan of Jello. Okay. Everybody's had or made Jello in their life before, right? Okay. Jello, if you take that pan and you shake it, what happens to the Jello? It wiggles, right? Okay. So like when you go from a head upright position and that Jello is nice and normal, and if you tilt it like a pan that's got Jello in it, look what happens to that membrane. Okay. The membrane shifts, and what's going to happen to those hairs? They're going to bend. Okay. And then that's what triggers those guys to depolarize, which then synapse with the sensory neurons, would form the vestibular branch. Okay. And those carry the signal into the pons and medulla. Okay. Now, very, very important for balance is information coming from your eyes, as well as signal coming from your joints from your proprioceptors. And we talked all about proprioception and proprioceptors, like your muscle spindles, tendon organs, and your joint kinesthetic receptors. Those are all different receptors that help you to maintain equilibrium or balance. Okay, so you have a lot of information coming into the vestibular nuclei, which integrates that information, then sends out signals that control movements of our eyes, help control head and neck movements, and control muscle tone in order to maintain equilibrium. Other neurons are going to carry signals up to the thalamus from our vestibular nuclei. The signals then from the thalamus travel to the vestibular area of the cortex, which is in your parietal lobes, providing us with conscious awareness of the position and movements of our head and limbs, not heads. We only have one. Now, as far as our last sensation, I'm going to tell you right now, this is the most complex, which you might be screaming inside right now because those last two sensations, hearing and equilibrium, were pretty dang complicated. But as far as vision or the act of seeing goes, I'm going to simplify it quite a bit. Okay. More than half of the sensoreceptors in your human body are located in your eyes. Think about that. All those different receptors that are in our skin, all those that are in our muscles, our tendons, our joints, in our ears, in our nose, they only add up to about half as many as what are in our tiny little eyeballs. Okay. And a very large part of our cerebral cortex is devoted to processing our visual information. Okay. Within our eyes are several layers, the most important of which is called the retina for the sense of sight. Okay. The retina is the layer where those receptor cells for vision are located. Okay. Those cells are called photoreceptor cells or photoreceptors, which absorb light and generate a chemical or electrical signal. Those include what we call rods and cones. You probably have heard of those before. If you haven't, well, now you have. Rods are responsible for night vision. That means they cannot distinguish colors from each other because when it's dark, you can't tell yellow from green or blue from black or purple from green, at least, when it's really dark. Okay. Each retina has a lot of rods, about 120 million of them. Okay. You also have what we call cones, function in bright light and are responsible for day vision as well as color. I always think C for cone, C for color. Okay. We don't have nearly as many cone cells as we do rod cells, about 6 million cones in each retina. Okay. Now these guys are both examples of separate cells. So they're not actual neurons, but they communicate with other cells in the retina that aid the rods and cones in vision. And those non-receptor retinal cells include bipolar ganglion cells and horizontal amacrine cells. Bipolar cells provide a large amount of convergence and stimulate ganglion cells. They're the ones that receive the signal from our rods and cones. We call them bipolar cells because they are bipolar neurons. Okay, and those are the blue ones here. Okay, looking at our illustration, we're zooming in at just this little tiny segment of an eyeball. Okay, each of our eyes, as far as the retina goes, has several layers. You can see those layers kind of broken down here: the pigmented layer, photoreceptor cell layer, bipolar layer, etc., etc. Okay, the names rods and cones come from what they look like. The rods have kind of a rod shape. Cones have a cone shape. Okay, those are found in the photoreceptor cell layer, which makes sense because those are photoreceptor cells. They become activated and then they synapse with the bipolar cells. Okay, bipolar cells then synapse with ganglion cells, and I would highlight these are the cells that form the optic nerves. Remember, the optic nerves are your cranial nerves number 2. Okay. There are also horizontal and amacrine cells. They're kind of sprinkled in here in the bipolar cell layer. Okay, horizontal cells, you can see there's one, kind of looks like an octopus. Here's an amacrine cell, kind of also looks like an octopus. Okay, but they form connections between other cells and enhance perception and contrast. They help with identifying the edges of objects and changes in light intensity. Okay, Now, what takes place is really cool. Look at how many rods and cones we have out here. And then look how many ganglion cells we have in here, okay? There are a lot more rods. There's a few less of those bipolar, amacrine, and horizontal cells. And then there's very few of these ganglion cells. This is creating what we call convergence, where we're going from a lot and we're funneling them down to a lot less. Okay? So that's what convergence is referring to. And that's what's taking place with these ganglion cells, which then form our optic nerves. Okay. So it all begins once again by creating a receptor potential in your rods and cones, which then releases neurotransmitters that creates greater potentials in the bipolar neurons and horizontal cells. And then signals are transmitted to the ganglion cells, which help form the optic nerves. Now, what takes place next is really crazy. It's kind of like what we learned when it came to what happens in our medulla with those pyramids. What we call the optic chiasm is where the two optic nerves, one coming from each eyeball, they crisscross. Okay. Now, does that mean your right eye, everything you see in your right eye is processed in the left brain? The answer to that is no. Okay. If you just break down each eye, there's two halves, a nasal half and a temporal half. Okay. See that over here with the left eye. Okay. Nasal half closer to the nose, temporal half farther from the nose. Okay. What we see in the temporal half does cross over. Okay. Whereas with the nasal half, it does not. So when you look at with one eye, for example, you're processing what you see in both sides of your brain. So when you're looking with both eyes at the same time, like we typically do, you have signals going all over the place in your brain. Ultimately, most of that signaling is ending up in the primary visual area of the cerebral cortex, which are in your occipital lobes, back of your brain. And same thing, don't worry about all the ins and outs of what's going on. Definitely make sure you know the different cells and what they do when it comes to vision. But I'm going to keep it really, really simplified, which is for the best, because like I said, vision is the most complicated of all of our sensations, and rightly so. It's pretty darn amazing. All right, any questions on sensations? Okay, well then with that said,